titled Population Dynamics and Management of Pepper Weevil on Jalapeno Pepper in South Florida. And just a general overview, I'll be giving a brief overview of pepper. I'll talk about pepper weevil, which is the main pest I'll be talking about today. Talk about few current management strategies, some problem areas, and talk about some of my research objectives for my PhD program, materials and methods. And just to start with, I'll give a brief overview of pepper, which belongs to the genus Capsicum annum and the plant family Solanaceae. Pepper is one of the 5 most cultivated species in tropical and subtropical regions of the world. We have the sweet and the hot pepper varieties. We have the bell pepper, and we also have hot pepper varieties, including jalapeno. And jalapeno is a common hot pepper to cook with. It has a mild to moderate pungency, which ranges between 1,000 to 8,000 Scoville each unit. Jalapeno is a perennial crop, and characteristics such as its pungency, flavor, aroma makes it an important ingredient in people's daily diet. Jalapeno is usually harvested as green, mature fruit for fresh market pepper. So we have constraint on the level of production, which include attack by pests and diseases. All of this I have here are common pests of pepper, but then the most important here is the pepper weevil because it is difficult to manage once it becomes established in the field. We also have some other group of insects in the Cuculionidae family, which pepper weevil belong to. Here we have the pepper weevil itself, we have the boll weevil, we have the hibiscus bud weevil, we have the alfalfa weevil. Although it's not antinomous, the genus is Hypera. We also have the strawberry bud weevil and the Cuban pepper weevil. But today I'll be focusing on pepper weevil. And it originated from Mexico, and it was first reported in Florida in 1935. Its primary host plants include flower buds, fruits, and seeds of plants in the Solanaceae family, which is mostly in the genus Capsicum and Solanum. It's important to mention that all pepper varieties are susceptible to pepper weevil attack, and both larval and adult feeding causes bud drop. We understand that the populations are high in locations across Southwest Florida region. As we can see on this maps, the red area is the red color is a region where pepper weevil population is high, and in Central Florida we have the yellow color. And the total production value of Florida fresh market bell pepper was 235 million, which accounted for about 45% of US value. And United States is one of the countries with the highest consumption volumes of pepper in 2018 after Vietnam and India, and with a market value of $374 million, while the global average yield per hectare was 101.3 ton in 2018. So talking about the life cycle, the female lays a single egg in the fruit wall. Once the egg hatches into larva, it moves into the fruit and starts feeding on the fruit tissues. It passes through all the immature life stages inside the fruit. So when it becomes an adult, it makes an exit hole on the surface of the fruit, and it comes out through this exit hole. And life cycle is about 15 to 22 days, depending on temperature and other food resources available. Here we can see in this picture to the right, the larva, 2 larvae and 1 pupa. So here are various management tools for pepper weevil control, and I'm going to go through some of them in the next slides. The first one is monitoring, and we know that successful management begins with diligent monitoring, which is usually aimed at early detection. And one of the most reliable tools that we use is the use of a pheromone trap, which often consists of a yellow sticky card and pheromone lures. The male pepper weevil produces aggregation pheromone, and studies have been done to identify the aggregation pheromone for pepper weevil, and it's being sold commercially. These lures, they contain some chemical compound that attract both males and females. And some of the importance of using the aggregation pheromone is to help detect infestation early and also help to improve the ability to manage pepper weevil and reduce crop losses. We also have chemical control, which is a major primary tool, and this includes the use of organic, conventional, and reduced risk insecticide. We have Actera and Vidate, which are two major insecticide growers used in South Florida. We also have a new insecticide, which is from Syngenta, which is called Plinazoline, and it's also effective for pepper weevil management. And we understand that conventional insecticides are all vital for managing pepper weevil, but even a weekly control plan with pesticide rotation cannot completely control pepper weevil. Despite the presence of all of these chemical insecticides, some losses are inevitable. And also, the repeated and heavy use of some of this insecticide has led to the development of resistance and the elimination of natural enemies, and the need to develop other alternative approaches that help to reduce the fast development of resistance, or at least new strategies that we can incorporate into IPM programs. We also have cultural control that help to limit initial immigration into the crop, and these are usually helpful to reduce or manage the population level during the crop cycle. One of the cultural control is the removal of alternate hosts, removal of old pepper plants, and white solanaceous hosts from the field. We also have a crop-free period for food deprivation. Also, we've used manual collection and destruction of fallen infested fruits. The use of mesh screens covering vents in greenhouses has also helped to reduce the movement of pepper weevil in and out of the greenhouse. And also intercropping pepper plants with plants that served as barrier crops help to reduce pepper weevil population compared to the crops, compared to the plots where intercropping was not observed. So also we'll have biological control, which is an alternative strategy that we are trying to incorporate into IPM programs. However, little is known about the natural enemies of pepper weevil, but in Mexico, two parasitoids have been observed attacking pepper weevil, which is the Triaspis and the Catalochus. And in Florida, the picture here, the one below is the Catalochus, which is the most common parasitoid and the most abundant in Florida, which parasitizes the third instar larval stage. Although it was reported that there is difficulty for the parasitoid to access the third instar larva due to the development of the immature within the fruit. And in the lab, we have a colony which is being read by one of our master students in our lab, and she's doing some studies. That's Nagamani. She's doing some studies with these parasitoids. Also, I need to mention that pepper weevil is limited to plants in the genus Solanum amcarsicum, and all of the 5 domesticated species are suitable for oviposition and development. Early infestation in pepper field is usually due to the presence of alternate host or old pepper plants growing around the pepper field. So therefore, it's important to determine strategies that can help to delay migration of pepper weevil from alternate host to its main host, since we can't 100% control this factor. And so with all of these tools I've mentioned, we still see a need to understand pepper weevil population dynamics, be able to differentiate between sexes, and identify other alternative strategies that can help to delay infestation and also increase pepper yield. Pepper weevil can be present before flowering and even until after harvest, but growers usually detect pepper weevil infestation when the fruit begins to drop. And at this point, it's already late to have an effective control because the pepper weevil population has already been established. And only adults are actually exposed to the product applied. And some of these products have, pepper weevil have developed resistance to some of these broad spectrum insecticides. Heavily infested fruit or fields can lead to 100% yield loss, and due to early infestation and lack of proper management strategies. So it's important to understand the biology, behavior, and the spatial distribution of pepper weevil for successful management. And yeah, some of my objective is to develop alternative management strategies to the solely use of insecticides. So for all of the reasons I've stated, my research is subdivided into three main objectives. I'm not going to talk about all of them today, but I'm just going to mention a few of them. And so the first one was actually to sex pepper weevil and identify morphological differences between the sex. So because identifying pepper weevil males and females is actually very challenging, and to be able to distinguish between the sex is important for sex-related studies. And also in order to use the live insect for lab and field-related experiments, it's so impossible to dissect the genitalia because we need to understand the markable differences between the sex. And I need to be able to do this for some of my field-related study and also some lab studies using olfactometer. So for this experiment, I collected pepper weevil infested fruits from pepper field. I brought them to the entomology lab and I put them in emerging cages. So after emergence, mating pairs were placed in a 5 millimeter Petri dish, and I was able to dissect them. And based on some previous known characters, and some efforts were also made to recognize new and discernible differences in the structures of the male and female. So after observing some of these differences, those differences were used as references for live insects. So we use the Leica wild microscope and the Keynes digital microscope to observe these differences in the lab. And also I was able to observe the adults under the microscope, and I was able to measure the different body lengths and weights, antennal segments, and all of the above mentioned characters or body parts. And at the end, I average across all of the insects to determine the difference between the sex. And so to confirm sex, 50 live adults that are considered or that were considered male and female after observations were marked, and I kept them separately. And then I dissected the genitalia to confirm the accuracy of the sex identification that I observed. And what I did was actually getting 50 vials like this, as you can see in this picture. Then I identified them as male and female. I had to do this twice. So I have 100 insects in total. And out of the first one that I did, I missed 3 out of 50, while 2 out of 50 were incorrect for the second one I did. So I was able to identify at least about 96% of all of the insects that I dissected. And also to confirm the sex, I was able to take a male and female pair, place them in a 50-meter vial, wrap them around pepper leaves, and covered with paraffin to confirm oviposition after 3 to 4 days. And from some of the results I had, this is the average across 10 insects. We could see a difference in the rostrum lengths. As we can see, I'm not sure if this picture is clear enough, but the rostrum length in the female is a little bit bigger and longer than in the male. Also, the insert of the antennae to the end of the rostrum was longer in the female than in the male. And also the width of the last tegite here in this picture, this is a male and this is a female. For the female is shorter, while in the male it's kind of longer, as we can see from these pictures as well. And this is the difference between the male and the female. The males have a darker projection, as we can see in this picture, at the base of their tibia, and this extends out compare as in the female. We can't really see the projection in the female, but in the male, it's more obvious and it's more prolonged. So moving on to my second objective was to determine the population dynamics of pepper weevil under field conditions. So the first one, sub-objective under objective 2, was to determine pepper weevil dispersal distance using the mark release recapture method. So this study was mainly to evaluate how far pepper weevil can perceive its commercially made aggregation pheromone, which we can see on this trap. The black rubber tubes is the aggregation pheromone, and then the yellow sticky traps to be able to catch or trap the adult pepper weevil. So I had a center a center place where I released the insect from, and then I had 4 traps on each of the side, north, east, west, and north, east, west, and south of the field. And then I did this at different distances. The highest distance I was able to do was 200 feet before I left. And I think I'm going to continue to take this. I think I'll continue the study in the fall when I return to Florida. And this picture was taken actually when the traps were at 5 feet distance from the center point. And at the time of release, I would take the wind, the pressure, and the temperature. They were recorded for each of the release. And I had 7 releases for each of the distance. And at each time I release, I had 50 insects marked. So just a short summary of the results I've gotten from the study. I was able to capture more adults in the 5 feet distance compared to the rest of the distances, but at least at 200 feet, I was still able to get some adults that were trapped on the pheromone trap. So this is to tell that even at 200 feet, they would fly from wherever they are or wherever the non-host plants are. And although I still have to analyze the data to also confirm if there's any correlation among all of the variables that we are putting into consideration, including temperature, wind, and pressure. So the next sub-objective under objective 2 was actually to monitor pepper weevil abundance, still using the pheromone-baited sticky trap. And this information will also help us to be informed about pepper weevil behavior and abundance across the seasons. It also helps to understand the importance of host plants and alternate hosts when the hosts are not present. So for this study started January 2022. It's going to continue till December 2023. It's still ongoing. And at the beginning, I had 3 traps placed at different places at Tropical Research and Education Center. One is an open grassy field. The second one is a mixed vegetable field. The third one is also a mixed vegetable field, which is in block 9. And the other one is in block 3. And then few months ago, I had to place another one in the forest area where we think the non-hosts may be present or not. Just to have an idea of what's going on there in the forest area. So every month we replace the trap, but sometimes the pheromone traps are changed bi-weekly depending on how dirty the traps are. And at each time, the temperature is recorded, and also we are putting into consideration the presence or absence of the hosts around these areas. And the hypotheses were that pepper weevil would not be abundant across all seasons. And we hypothesized that temperature will influence pepper weevil abundance, and pepper weevil number will be lowest or absent in the open grassy field. So at the end of every month, or at the end of the study, we're going to compare the total number of adult per trapped amongst field and across seasons. And the data will be analyzed using GLM. But I was able to make a graph of what I have so far. And from last year, we could see that the flight activity was bimodal. We had a peak year between February and March 2022. and another peak here between May and June. And we are observing like the similar pattern this 2023. And before I left, I had a peak here, and Nagamani is going to help me take the rest of the data in the next 3 months until I get back. And I don't have that data yet for May, but I'm going to add it, and probably by the time I defend, we'll be able to see the results of the study, and we'll be able to compare the differences, or if there's any positive or negative correlation between both years. So my third objective was to evaluate pepper weevil under different management regimes. And the first one here is to evaluate the effect of combining reflective mulch and standard insecticide on pepper weevil suppression. And the second objective was to evaluate the effect of intercropping non-host and insect repellent plants with jalapeno pepper on pepper weevil suppression. So because we cannot rely on one method of control, although we know the major tool is the use of chemicals, we still need to integrate different control strategies for effective management of insect pests, which need to be not only cost effective, but as well as less harmful to beneficial insects and the environment. So yeah, we wanted to evaluate the impact of this metallized reflective plastic mulches, because they've been known to reflect ultraviolet light and repel insects by influencing their vision and locomotory behavior. And it's been effective against thrips, whiteflies, and broad mites. And Razak et al. 2018 also conducted a study and to evaluate the combined effect of reflective mulch and natural enemies on thrips farming. And also we've not had any previous study that evaluated the effect of reflective mulch on pepper weevil, but it's been used on bell pepper and jalapeno pepper against melon thrips, and the results were actually impressive. So that was the reason why we wanted to try it against pepper weevil suppression. And this study was conducted at TREC. The experimental design was a split pod design. It was done in fall 2020, spring 2021, and spring 2022. As you can see here, we have the 3 different mulch types: the regular mulch, the reflective silver and white mulch, and the no mulch. And we also had 2 subplots, which were the insecticide treatment plots. We had the control, which represents C here. We had the Actera and the Vidates. This Actera and Vidates are the 2 local standard growers used in South Florida. So we have published the results of the study. And if you have any question or you want to see the results, you can check this paper or this article online. But just to give a brief overview of the results, reflective silver and white mulch has the potential in reducing pepper weevil population and increasing pepper yield. We also saw that rainfall and temperature are major factors that speeds up mulch deterioration. And we also noticed that reflective mulches are more effective during periods of little or no rainfall. And so the next objective I'm going to talk about was to determine the effect of non-host and insect repellent plants intercropped with jalapeno pepper on the abundance of pepper weevil. So for this study was also conducted at TREC. We had 6 treatments, and each of the plot size were 15 feet long. And the experimental design was randomized complete block design. So as we can see in these pictures, we had pepper in the middle, while we had the non-host. Here is the cabbage on both sides of the bed. We had cabbage, we had basil, we had marigold, we had cilantro. We also had a control treatment where only peppers were planted, and we had another treatment where we sprayed Actera. So the number of pepper weevil adults and infested fruits were counted per treatment plots in this study, and at the end of the season, marketable fruits were collected, counted, and weighed. So all of our seeds were obtained from Aris and Semini seed. We planted the seedlings in seedling trays in the greenhouse using pro-mix soil. And after 6 weeks, the plants were transplanted. The results from that study has also been published. In case you want to check, you can see it here. It was published in Crop Protection Journal. But although we had some challenges here during the period when we conducted those experiments, and the challenges were competition as competition between the non-host and the main plants. As you can see in the cabbage, the plants were smaller compared to these other treatments where we had the plant intercropped with marigold or jalapeno just with Actera spray. And also we had issues with the plants in this cilantro intercrop plots. They were smaller compared to the rest of the plots. So because of these challenges, we decided to repeat this experiment where this study was done in 2023, where I had the non-host plants on separate beds. As you can see here, in addition to the non-host plants, I also wanted to evaluate the combined effect of the reflective mulch together with the non-host plants. So I had 4 repetitions. In this picture here, we can see I had the non-host plant, for example, in this case, marigold, on 2 rows, then pepper in the middle with either the reflective silver and white mulch or reflective silver and black mulch or the regular mulch type. And then on this other row, we also had 2 rows of marigold. And this is a bigger picture of the plot where we had marigold, cilantro, lemongrass, basil, and we had a control treatment without insecticide spray, and then we had a treatment with insecticides. So for the data collection and the evaluation of treatments, we considered the number of infested fruits per treatment plot, and the fruits were also collected, counted, and weighed at the end of the season. We will analyze the data using ProGlimis, and the means will be separated by Tukey's HSD tests. Although the results of the study is yet to be analyzed, but hopefully I'll be able to present them like when next I give a talk. And for my future study and plan, I plan to repeat the study again after analyzing the data from this last spring study. I plan to complete my mark release recapture study greater than 200 feet. My qualifying exam is coming up this fall. My dissertation exam, the writing and my defense, and hopefully I'll be able to graduate summer next year. And with this, I would like to conclude my talk. And first of all, I'd like to acknowledge all of my committee members, especially Dr. Sil, my supervisor, for their continuous support, advice, and guidance, and even for allowing me come for this internship. It's really a great opportunity, and I'm so excited for all the new things I'm learning. And yeah, I also want to thank everybody in my lab for their help and support with data sampling, planting, and all they do. I also like to thank James Coley, which he's been the one helping with the statistical analysis, and also pepper growers in Miami-Dade County for allowing us sometimes use their field for experiments and other things. And to everyone present today, thank you for listening.